Hello, I'm Alex, host of AI Research Roundup. Today, May 7th, 2025, we're looking at an ICLR conference paper, Learning and Aligning Single-Neuron Invariance Manifolds in Visual Cortex, published a few months back, January 22nd. So, this paper tackles comparing how visual neurons show invariance. That's consistent responses to varied stimuli. It learns a neuron's continuous invariance manifold, its preferred stimuli set, then aligns these across neurons for better comparison. Alright, so this figure 1, really lays out their two-step approach. First is template learning, where they use an implicit neural representation, or INR, to learn a continuous set of images that a single template neuron really likes. Then, in template matching, they use an affine transformation that's like a geometric tweak to align this learned set to a target neuron, aiming to maximize its response as well. This helps compare them directly. Okay, so figure 2 puts this method to the test using simulated neurons. Panel A shows various Gabor-based neurons, artificial ones with known ground truth invariances. Panel B then shows the manifolds learned by their system. Crucially, panels C and D display an activation matrix, before and after hierarchical clustering, clearly grouping similar neurons. Panel E confirms high accuracy against this ground truth, validating the learning and alignment. Building on that validation, figure 3 now applies this to actual macaque V1 neurons. That's the primary visual cortex. Panel A displays learned invariance manifolds, grouped by clusters identified in Panel B's activation matrix, which uses hierarchical clustering. Panels C and D show consistency across different experimental initializations, called seeds, and even across animals. Panel E further demonstrates that these findings generalize well to different predictive models, which is quite robust. And for those listeners keen on digging deeper into the methodology, this table of contents from page 14 points to supplementary sections. It details things like MEI generation, that's maximally exciting input generation, template learning, the simulated neuron setup we saw in figure 2, and even the macaque response predicting models. It's where all the nitty-gritty experimental details live, supporting the main findings we've discussed so far. Okay, so diving deeper into those supplementary details, Figure S1 on page 17 explores how defining near and distant latent points affects learning. Panel A shows a ground truth manifold for a complex cell. Then, C varies these proximity definitions for contrastive learning. Panels D and E show how the learned manifolds and their smoothness change. Panel F confirms that learned manifolds still elicit high neural activations, showing some robustness. Alright, turning now to figure S7 on page 23. This one explores the impact of using different underlying predictive models. Specifically, they compare a standard CNN to a ROT EQ CNN. That's a rotation equivariant CNN, good for handling rotated inputs. Panels F and G show their clustering method still works well for both, demonstrating the robustness of their overall approach across different neural network architectures for predicting responses. Following up on how different models perform, Figure S3 on page 26 looks closer at their predictive accuracy. That's how well they predict actual neuron firing. Panel A shows Model 1 maintaining good correlation across all identified neuron clusters for both macaques. Panel B then pits Model 1 against Model 2, a different architecture, revealing very similar predictive performance. This consistency is key, ensuring the learned manifolds are based on solid neural response predictions. So, this consistent predictive accuracy is really foundational. What we've explored is a novel framework for characterizing neural invariance manifolds, essentially how neurons generalize. They learn these using implicit neural representations, or INRs, predicting responses with deep neural networks, or DNNs, and then align these manifolds across neuron populations in V1, the visual cortex, to find shared computational roles. This really advances our understanding. Thanks for tuning in.